So I'd like to thank the committee over here for inviting me to come and participate in the Oliver Smithies Symposium. This is such an honor for me for multiple reasons, in part because I get to give a lecture that's named after Oliver Smithies alongside with Professors Jody Nunari and Doug Wallace. So that alone is like a huge honor. But number two, being able to come to Madison, Wisconsin, which has such a tradition in mitochondrial biology and bioenergetics, to be able to come here and give a lecture on this particular area where many fundamental discoveries were made is quite an honor. And finally, and perhaps most importantly, because I really do get to come back and visit Dave and his remarkable laboratory. This morning I was just blown away by seeing him and his relatively young group making such advances. And so I feel really honored to be here today. So I'm going to share with you some of the work we do. I'm going to provide an initial overview of the work, of our approach, and then I'm going to dive deep into one problem that's been of deep interest. And throughout my talk, as you will see, there are some very important links to University of Wisconsin. So the theme this year, of course, is mitochondria. I'm going to provide a very, high-level overview of this organelle that should be obvious to most of you. So these are obviously ancient organelles. They're found in virtually all of our body's cells. They're constantly undergoing fusion and fission, which we'll hear about later. And under light microscopy, they actually look like intracellular bacteria because, of course, 2 billion years ago, that's the far right panel, these were free-swimming bacteria, probably resembling modern-day gram-negative rods, something like a Rickettsia prowazaki. And they still maintain a double membrane structure and still perform many of the functions that bacteria do. And so today their major functions include things like energy metabolism, redox balance, ion homeostasis, and over the last decade, I would say there's growing appreciation for the role of this organelle in signaling and in cell death. Our lab is also very interested in a very large collection of individually rare disorders. These are the orphan mitochondrial disorders. They impact about 1 in 5,000 live births. They're characterized by a deficit in the oxidative phosphorylation machinery. This is the primary pathway by which our cells generate ATP. And they're incredibly challenging because of the very large number of genes that can be involved. So there's over 150 distinct genetic forms of these disorders. And there's also a lot of pleiotropy. There's a lot of phenotypic heterogeneity. So in some of these disorders, all you will see is blindness. And in some of these disorders, you'll see blindness, you'll see a subacute degeneration of the brainstem with an anemia and a myopathy. And at present, why certain lesions to the mitochondria give rise to multisystemic disease, whereas others give rise to highly tissue-specific disease, is a beautiful unsolved problem in the field. And there's growing interest and excitement in mitochondrial biology because it's quite clear now that many common age-associated disorders are associated with probably a quantitative decline in mitochondrial activity. Almost all tissues as they age exhibit a decrease quantitatively in mitochondrial amount, in mitochondrial activity. And whether that's just reflective of the aging process or actually contributing to the aging process is an unsolved problem right now. In a small subset of cases, it's quite clear that this can actually drive the pathology. So one of the landmark events in our understanding mitochondrial biology and mitochondrial disorders was the sequencing of the mitochondrial genome. And this is an amazing, amazing paper, and I really encourage all of the students here to go back and read this paper. This is actually a fascinating issue of Nature back from 1981. So this was published almost 20 years, almost exactly 20 years before rest of the human genome would be sequenced. And the author list is pretty distinguished. It includes people like Fred Sanger, who is listed in the middle of this author list because he actually preferred for the author list to read alphabetically. It included one of the first bioinformaticians, somebody named Staden, who I think some of you will actually be familiar with. And there's a couple of companion articles in the same issue. In a lot of ways, this was actually first-generation sequencing. This was first-generation DNA sequencing. And in the same issue of Nature, there were companion papers that attempted to annotate the mitochondrial genome with respect to its gene products. And thanks to some of these early studies, we actually know that the mitochondrial genome encodes a total of 13 proteins. It also encodes 2 RNAs and 2 rRNAs and 22 tRNAs. These are all the RNA components necessary to translate those 13 proteins off of the mitochondrial genome. So this is a landmark event in our history of this organelle. But of course, mitochondria do many other things, and all of those other things are not encoded by the mtDNA. And so if we just take a look at, I don't know if we have a pointer, or if we could get a pointer. I have a pointer. Let's see. Oh, thank you. Oh, I got one now. Thank you. Okay, great. Okay. So this is a reminder of sort of the mitochondrial central dogma. So the mtDNA encodes 13 proteins. These are all core subunits of complexes 1, 3, 4, and 5. And then 77 subunits of oxidative phosphorylation are nuclear DNA encoded. And so obviously the nuclear genome is immediately of importance because there are 77 subunits even for the OXPHOS system that have to get imported. But even all of the protein components necessary to fully express the mtDNA are nuclear genome encoded. So there are numerous factors, nuclear encoded, that are required for the replication and maintenance of the mtDNA, for its transcription and for the RNA processing, for the translation, for the import of these subunits, and for the full assembly of oxidative phosphorylation. So even when we just restrict ourselves to oxidative phosphorylation, it's quite clear that if we really want to have a systematic understanding of this organelle, in addition to the mtDNA and its components that it encodes, we need to consider rest of the cell. And as I was completing my clinical training in 2001, rest of the human genome was coming online. This particular paper represents the culmination of the public consortium's efforts to sequence the human genome. Much of this came from different Cambridge, Massachusetts, and was being directed by Eric Lander at that particular site. So I ended up doing a postdoc in Eric Lander's lab, joint with Matthias Mann, and my goal was to try to understand what the nuclear genome's contributions are to mitochondria. And in some initial studies, the goal was to try to computationally predict all of the mitochondrial proteome. We had a few papers attempting to figure out which of the 20,000 proteins manufactured by the nuclear genome end up in the mitochondria. And so that was a decent first guess using computation. And then we began to develop various mass spec methods to try to identify proteins that are found in purified suspensions of mitochondria. But then these efforts really took off when we were able to launch our own laboratory, and I was able to recruit Dave Pagliarini to the laboratory. And this was our efforts starting in 2005, when Dave started in the lab, to really try to map out the mitochondrial proteome, really to try to come up with a molecular blueprint for this organelle. And what Dave did in collaboration with Sarah Calvo, a computational biologist in the lab, and in collaboration with Steve Carr, our proteomics collaborator, was to basically map out all of the proteins found within mitochondria from 14 different mouse tissues. And so we purified mitochondria from 14 different mouse tissues, with the idea that the protein composition might be different from tissue to tissue. We developed some biochemical methods to try to distinguish genuine mitoproteins from abundant contaminants. We developed methods with which to analyze all of the mass spec data using a Bayesian approach. And we were then able to score all proteins in the proteome for their likelihood of encoding a mito-targeted protein. And in parallel, we did a high throughput GFP validation study, and in the end, we're able to put together an inventory of about 1,100 proteins that we called mitocardia. And this was actually still remains probably one of the best and most comprehensive catalogs of the mitochondrial proteome. We included 14 different tissues, and so we also reported information on the relative abundance of these 1,100 proteins across 14 different tissues. About 600 proteins are really unstudied. Some of these proteins have domains that would actually hint at what their function is, but we really don't know what they do. And so depending on what your metric is or what your criteria is for well-characterized or well-studied, this number ranges anywhere between 300 and 600 proteins. And so this particular inventory has been extremely valuable to the field. Dave's paper, I think at this point, has been downloaded. The data set has actually been downloaded over 10,000 times at this point, and serves as a molecular blueprint for lots of people's work in the field. And we continue to use this inventory in our laboratory. And so we use it in two ways. One is we've used this inventory to guide our search for Mendelian causes of mitochondrial disease. And so when we see patients that have clinical phenotypes resembling those with mutations in the mtDNA, but the disease is segregating in a Mendelian manner, we can do sequencing of the mitochondria genes and identify, in many cases, recessive causes of the disease. Now in the era of exome sequencing, we do exome sequencing followed by prioritization of the mitochondria genes. And so that's been a big focus of ours over the last couple of years in the post-mitochondria era, if you will. But today what I want to do is I want to share with you a story that builds on mitochondria that's very basic in nature. And this particular story actually traces its origins back to Madison, Wisconsin. And so more than 50 years ago, right when some of the principles of chemiosmosis, chemiosmotic coupling were being discovered, multiple groups at Johns Hopkins, at the Johnson Laboratory in Pennsylvania, as well as over here in Madison, namely the work of Professor DeLuca, in whose honor I think mall buildings here are now named. I actually don't know how many of you know this particular contribution from him, but he and others discovered at that time that if you purify mitochondria and you energize them and you give them a pulse of calcium, they will uptake that calcium and buffer it. And so purified mitochondria from almost any source have almost an insatiable appetite for calcium. And that's shown over here in these classic traces. So you take mitochondria, you give them substrates, things like glutamate and malate. You also have to give them inorganic phosphate, and over time they'll accumulate huge amounts of calcium. And as it turns out, vitamin D doesn't matter too much for this particular phenomenon. This is also shown at Johns Hopkins and Albert Leninger's research group. If you give just purified mitos calcium, not much happens, but if you actually energize them, and you also have to give them inorganic phosphate, they'll uptake huge amounts, and it's believed that they actually buffer the calcium inside of the matrix as a calcium phosphate product. And over the next 50 years, since those early studies here and at Pennsylvania and at Hopkins, numerous groups have examined why calcium might matter. And there are three main roles for mitochondrial calcium that have been identified over the years. Now, the first is what's often called metabolic coupling. I like to call it bioenergetic coupling. And so the idea is that calcium will often rise in the cytosol to activate an ATP-consuming process. So something like muscle contraction or neurotransmission, these are both activated by a spike in calcium, but they're also ATP-consuming. So somehow or another, the ATP utilization needs to be matched by its production. And it's been proposed that calcium serves as a feed-forward signal. And that's because when calcium goes into the mitochondrion, there are three TCA cycle dehydrogenases. They're pyruvate dehydrogenase, oxyglutarate dehydrogenase, and isocitrate dehydrogenase. All three of these are allosterically activated by calcium. And so the idea is that if you want to trigger an ATP-consuming process, you have a calcium spike, and that same calcium goes into the mitochondria, boosts the TCA cycle, and this results in impedance matching between ATP production and utilization. So that's one role for mitochondrial calcium. Another role for mitochondrial calcium is in controlling cell death. And so these are more modern day versions of the initial DeLuca experiments. And so if you take purified mitochondria, you can incubate with them a particular reporter that's outside of the mitochondria, and that reporter will fluoresce in the presence of calcium. You give a pulse of calcium, you get a rise in the free calcium outside, and then the mitochondria will uptake the calcium and buffer it. And you can continue to give pulses of calcium. It's a very robust phenomenon. And at some point, you exceed the buffering capacity of mitochondria, and now the free concentration of calcium inside is very high. There's osmotic swelling. The mitochondria will pop. This is what's often called the mitochondrial permeability transition taking place. And so almost all forms of cell death are associated with calcium overload. An open question is whether mitochondrial calcium uptake is driving the cell death, or if it's simply reflecting a dead cell. And then finally, the third place where mitochondria and calcium are extraordinarily important is in the tuning of cytosolic calcium signals by the mitochondrion. And I think this is really nicely shown in a video from my colleague Jim Lechleiter from UT San Antonio. So I'm going to show you these two videos. What these are videos of Xenopus oocytes, and he's injected into the Xenopus cytosol a calcium reporter. And so we're not looking at the mitochondria. This is the cytosolic calcium that you're going to be seeing. And then he injects in IP3. So this mimics what happens soon after fertilization. You're going to see these waves of calcium. Okay? So the only difference between the left Xenopus and the right Xenopus is that here the mitochondria are energized, and you'll see what happens. So hopefully what you can appreciate is that when you inject an IP3 into a Xenopus, you get these calcium waves that have a low amplitude and a high frequency. Once you energize the mitochondria and they're allowed to uptake calcium, the tone is higher. Now the amplitude of these waves is higher and the frequency is slower. And so remember, we're looking at the cytosol, so the mitochondria can actually influence signaling outside of its domain via calcium. Okay? So three important functions: metabolic coupling, cell death, and the regulation of cytosolic signaling, all by mitochondrial calcium. Okay. So how does calcium get into mitochondria? And so this is the respiratory chain. And remember, these three proton pumping complexes within animals, at least, helps to generate a proton gradient and a pH gradient and a voltage. And this voltage difference is actually what helps to drive calcium uptake. So under resting conditions, the cytosolic free calcium concentration is very low. It's about 100 nanomolar, and we believe that it's approximately the same inside of the matrix. And under resting conditions, not much is happening. And now when you get one of these spikes of calcium outside, because there's a voltage difference, this calcium now flows down its gradient, and then it gets buffered. Okay? And so this transport occurs across a transporter in the inner membrane called the mitochondrial calcium uniporter. Okay? And this uptake occurs across the inner membrane of all vertebrate tissues ever examined. It's sensitive to a drug called Route 360. This is not a cell-permeable inhibitor, but in purified mitochondria or in permeabilized cells, this drug is a highly potent inhibitor of the uniporter. And David Clapham's lab back in 2003 was successful in patching mitoplasts. Mitoplasts are basically mitochondria in which you've removed the outer membrane. He was successful in patching the inner membrane of mitochondria, and he studied the biophysics of this transporter. And the transport kinetics are most compatible with a channel mechanism. And so this channel had been studied extensively for many decades, had been linked to a variety of cellular processes. But a major limiting factor in our sort of studies of this channel was that we actually didn't know what this protein was. What is the molecular identity of the uniporter? And this often happens in biology. So there's a different current called the crack current, which some of you are probably familiar with. They're very important in immune cells. That current was first characterized at a biophysical level in 1990, and it wasn't until 2005, 15 years later, that its molecular machinery, STIM1 and ORI, were identified. And so often, a channel can be studied without knowing what its molecular machinery is, and a situation was analogous over here. And so this is a problem that we've been wanting to take on for quite some time. I actually studied Uniporter biology and physiology when I was in Bob Balaban's laboratory, and we'd always wanted to try to figure out what the components are that encode this channel. And I'm going to share with you the work of multiple past and current members of the laboratory that decided to take on this problem. And so Fabiana Perocchi was the first postdoc to work on this problem. She now leads her own group in Munich. And Josh Boffman was a graduate student that, in a fortuitous and accidental way, ended up working on this problem. He's now a fellow at Genentech. And Molly Plavanich is an HST student, an MD student that was an MD student that's now doing her residency at the Brigham and Women's Hospital. Other members of my lab continue this work, including Yasmin Sanchek. She's a postdoc, extremely talented postdoc that's going to be going onto the job market this upcoming fall. And I want to make a shout out to Kim Kamer. She's actually an alumna of University of Wisconsin. She actually did her undergraduate studies here, and I think worked in Ron Rain's laboratory, if I'm not mistaken, and she has just done some really outstanding work over the last couple of years. Erica Kovacs is a postdoc, and Andrew Marquardt is a research assistant. So I'm going to share with you the work of this team of individuals. And so I introduced the talk with this mitochondria inventory, and if this is actually a decent inventory, then presumably the inventory must include the protein components of the mitochondrial calcium uniporter. And we actually assembled an RNAi library targeting all 1,100 proteins, and we were actually fully prepared to do an RNAi screen against mitochondria to find the component machinery. However, even before we did it, we decided to just do some back-of-the-envelope calculations. And so if we were to screen 100 genes and we set a p-value of 0.05, that meant that we're automatically going to identify 5 hits by chance alone. And if we screen 1,000 genes, that means we're going to get 50 genes by chance alone. But we do 5 hairpins per gene always. So that means that we're going to get 250 hits just by chance alone. And we haven't even picked up a pipette yet. And doing follow-up on 250 genes is incredibly difficult when we have to do biophysics and physiology. And so we decided, instead of diving in and doing a full-on genome-wide or organelle-wide screen, that we would prioritize with prior knowledge. And this is what we did. Now, we knew from the literature that the mitochondria of all vertebrates exhibit uniporter activity. And there's a really important paper in the early 1990s from Annabelle Versessi, who studied mitochondria from trypanosomes and from leishmania, and showed that the mitochondria from these highly diverged unicellular organisms also uptake calcium in a voltage-dependent way with pharmacologic properties that resemble that of the uniporter. So they're also sensitive to drugs like Route 360. So this suggested, it raised the hypothesis that trypanosomes and leishmania also have mitochondria that have the uniporter. The other key historical experiment that we're aware of came from Albert Leninger's group. So Albert Leninger had shown that if you purify mitochondria from yeast, they do not uptake calcium in any way that resembles that of vertebrates. And so these were really important clues that were basically in the literature. And in the era of computational biology, what we're able to do is we're able to ask which of the mitochondria proteins, which of those 1,100 proteins that Dave had helped to map out, have homologs in the kinetoplasts, but specifically don't have homologs in cerevisiae. And when we do this subtractive proteomics experiment, there are 58 proteins that have this phylogenetic profile. And so we ended up prioritizing those and went one step further and prioritized based on the proteomic analysis of Jean-Claude Martineau. He had actually characterized the mitochondrial inner membrane proteome from heart. And so when we crossed these lists that yielded about 13 proteins, we did an RNAi screen with 5 hairpins each against these 13 proteins. And we found one hairpin that was compatible with life, but gave a severe calcium uptake defect. We're expressing a reporter for mitochondrial calcium in the matrix. And so this particular hairpin really scored very high, and this particular hairpin corresponded to a protein called CBARA1 that had really not been studied before, and we ended up renaming it MCU1, so mitochondrial calcium uptake 1. So we're really excited about this protein. It has a mitochondrial leader sequence. It has 2 domains known as EF hands that are known to be calcium binding motifs. And EF hands come in 2 flavors. They come in high affinity and low affinity flavors. The low affinity flavors are important for buffering calcium, and the high affinity are important for sensing. These look like calcium sensing EF hands. And so the fact that this protein has the right phylogenetic profile, the fact that when we RNAi it, it gives calcium uptake defect, has these EF hands, got us really excited that we were really, really close to this Uniporter. And then when we did topology analysis and when we did carbonate extraction experiments, what we could show was that this protein is an intermembrane space protein. It's a peripheral membrane protein. It has no bona fide transmembrane domain, but it's a peripheral membrane protein that's on the outside of the inner membrane. And so what this led us to hypothesize is that what we had identified was probably some sort of a regulatory subunit of the pore. And so then this actually led us to ask the question, what encodes the pore? And this is where Josh Boffman jumped in. He was a computationally savvy PhD student working on a completely different project, which was not working very well, but he and Fabiana decided to team up, and they went fishing in a computational sense. They used MCU1 as a computational bait, and then they asked what other genes in the genome encode proteins that behave in the same way as MCU1. And so we performed 3 different experiments. The first is an evolutionary co-occurrence experiment. And here, what we did is we took 500 sequenced genomes, and then we compute the vector of absence or presence for MCU1 across the 500 organisms. And then we can compute that same binary vector of absences and presences for all other 20,000 genes in the human genome. And then we can sort all 20,000 genes based on the Hamming distance between the MCU1 binary profile and that of all other 20,000 genes. And so things on the far left appear and disappear with MCU1 in evolution. And then these almost have an anti-correlated expression across evolution. We can't say much about the genes in the middle. The next experiment we did was we asked, again, computation, was what other proteins within the mitochondria inventory have a similar pattern of expression with MCU1. So remember, we'd characterized the mitochondrial proteome across 14 different tissues, and so we can ask what the pattern of expression of MCU1 is across those 14 tissues, and then plot the correlation between it and all other 1,100 proteins. So these are mitoproteins that are strongly co-expressed across tissues with MCU1, and these are anti-correlated. And then the third analysis we did makes use of freely available microarray data. So the Novartis Foundation has generated a tissue atlas of about 60 or 70 different mouse tissues. They did genome-wide microarrays. All of this is in the public domain. And so we can ask which of the other 20,000 genes have RNA expression profiles across tissues that are correlated with MCU1. So these are 3 computational experiments. They take a couple of hours each to run. They're all aimed at determining what other genes potentially have unknown function encode proteins that are either co-evolving with or co-expressing with MCU1. And when Josh did this analysis, he identified a protein that was extremely high scoring. It was number 1 in this analysis. It was number 2 in this analysis, and tied at a score of 5 in the third analysis. So this is sort of the Michael Phelps of the analysis, sort of a gold medal, a silver medal, and a qualifier, which is pretty good. And so what is this protein? This protein is something called CCDC109A, and it has 2 predicted transmembrane domains, and this is exactly what you want in this type of analysis. I mean, the only thing that's known about this protein is that, across the entire genome, it's the 109th unannotated coil domain. And so really nothing is known about this protein, and the fact that it's an A means that there must be 2 of them, right, or at least 2. The only paper that had been published on this paper was actually Dave's mitochondria paper, and so it is a mitochondrial protein found in almost all tissues, and all of the computer programs predicted that this has 2 transmembrane domains. And so this led to the hypothesis that CCDC109A encodes the Uniporter, and we would end up renaming this protein MCU for reasons that'll be clear shortly. So I'll refer to this protein as MCU, and it's not to be confused with the first protein, MCU1. And so the computation predicted some sort of an interaction, some sort of a functional link between MCU and MCU1. So Yasmin Sanchak, after she started in the laboratory, expressed a flag-tagged version of MCU and could show that we could pull down MCU1, but not other mitochondrial intermembrane proteins. So there seemed to be a specific physical association between MCU1 and MCU. We worked with the pharmaceutical company Alnylam, which had been developing highly potent, highly specific siRNA reagents for in vivo delivery. So as many of you know, if you actually do a tail vein injection of siRNA into a mouse, that siRNA will get sequestered primarily by the liver, and with their reagents, we can actually achieve a remarkable knockdown of a gene of interest. So they generated for us MCU siRNA, and when we purify these mitochondria, I think many of you are probably familiar with state 3, state 4 measurements. Oxygen consumption is fine in the presence or absence of MCU. The membrane potential transitions are fine. So this means actually that they're doing oxidative phosphorylation quite fine. And if you give a pulse of calcium to normal mitochondria, this is very similar to the DeLuca experiment, then energized mitochondria uptake the calcium. However, in the absence of MCU, they no longer uptake calcium. So this strongly suggested that MCU is functionally important for mitochondrial calcium uptake. So recall that one of the proposed functions of MCU is metabolic coupling. So when calcium goes into the matrix, it activates the TCA cycle. And when you get activation of the TCA cycle, one readout of it is increased NADH. And we can actually monitor NADH levels using microscopy. NADH absorbs at about 380 or so, and it fluoresces at about 450. So we can actually monitor NADH levels really nicely in permeabilized cells. If you give them a pulse of calcium, there's an increase in NADH that is sensitive to this drug called Route 360, which is not cell permeable, but it's an inhibitor of the Uniporter. And so in a permeabilized cell, it blocks calcium uptake. And silencing MCU phenocopies treatment with Route 360. And so this suggests that MCU and this protein is very important for metabolic coupling. So MCU and MCU1 functionally interact. If we lose MCU, then we lose metabolic coupling. We also lose mitochondrial calcium uptake. We did some experiments to figure out the topology of this protein. So there's a carbonate extraction experiment that shows that this is a bona fide membrane protein. And our topology analysis suggested that this is an N-in, C-in configuration. So this protein only has 2 transmembrane domains, a very short linker, and our topology analysis places both the N and C termini in the matrix. Okay? Now, if this is going to be the pore-forming subunit of the Uniporter, then if it only has 2 transmembrane domains, it's hard to imagine how it can operate on its own. And so the hypothesis is that it must oligomerize very much in the same way as other ion channels. And we used either a V5 or a GFP tag on MCU, and if we tug on one, we get the other. So clearly it's forming at least a dimer, and this is also consistent with what happens when you run purified mitochondria on a BN page. If we take mitochondria from HeLa cells or from liver cells, the Uniporter runs at a higher molecular weight complex. If we eliminate MCU, absolutely nothing happens to the F1F0 ATPase. And as you can see, our RNAi reagents are pretty good. And so this is running at a higher molecular weight complex, suggesting that MCU forms at least a dimer and oligomerizes into something bigger. Okay. So we wanted to try to garner additional evidence that this really is the pore. And if we look at the sequence, there's a lot of acidic residues here at the putative opening of the pore. And Dick Chen and Patrick Eleanor had shown many decades ago that all voltage-gated calcium channels have acidic residues that are very important for things like selectivity and conductance. And fact, if we RNAi MCU and then reintroduce alanine mutants at any of these acidic residues, we actually abolish mitochondrial calcium uptake, consistent with the notion that these acidic residues are important for calcium transport. Now, we got very lucky at this point because we decided to mutate another residue in this vicinity just to show that there's something special about these glutamates and aspartates. And so Josh decided to also mutate the serine residue. Okay? As you can see, if we create an alanine mutant of this particular residue, mitochondrial calcium uptake is still intact. Okay? And whenever we do these mitochondrial calcium uptake experiments, we always want to make sure that we actually are looking at the Uniporter current, which is defined by its sensitivity to root 360. And so, so here's what Josh did. If we take cells that are expressing nothing, or the wild type, or the S259A mutant, if we give a pulse of calcium, they uptake and buffer it. But then we can add root 360 to make sure that the current that we're measuring is actually sensitive to this drug. And what we could see was that if we express nothing or the wild type, we still have sensitivity to root 360, but this S259A mutant is very resistant to the action of this drug. And in classic sort of biochemistry, if you want to prove that you've identified the target of a particular drug, one of the strongest pieces of evidence is to identify a drug-resistant mutant. And so this doesn't fully show that this is the direct target of root 360, but it shows that we're pretty darn close to its target. And we're able to do experiments in collaboration with David Clapham's lab to show that the Uniporter current that he originally identified in 2003 is fully ablated when we silence MCU. So this is the current that he first described in 2003 with his mitochondria mitoplast patch clamping studies. And this is the current that's sensitive to ruthenium red or root 360. And what you can see is that when we silence MCU, we lose this current. And in the same paper, I'm not showing the data, we could also show that this S259A mutant is completely resistant to root 360. And so, so we were able to accumulate quite a bit of evidence that this protein, CCDC109A, does indeed encode the pore-forming subunit of the Uniporter. So what's the evidence? It's MCU is required for mitochondrial calcium uptake in cells as well as in vivo in mouse liver. It's found in the mitochondria of all vertebrate tissues evaluated. And so in the original mitochondria paper, it's actually found in 12 out of 14 tissues. If you go back to the other 2 tissues that didn't have it, by Western blot, it's there. MCU has the expected phylogenetic profile that we first defined, namely present in vertebrates and kinetoplasts, but absent in yeast. MCU has 2 transmembrane domains and oligomerizes, and mutations at these evolutionarily conserved acidic residues abolishes calcium uptake. And we've identified a drug-resistant mutant for this pore. And finally, mutations in MCU alter the I-MICA current, the current that David Clapham identified as being the signature current for the Uniporter. And so soon after we made this discovery, around the same time, Rosario Rizzuto's group reached similar conclusions. I think together helped enforce the idea that this second component that we've identified is very, very important for the Uniporter. And so, so this is where we were a couple of years ago. So MCU1 appears to be the soluble protein in the intermembrane space. It's a peripheral membrane protein that appears to be important for the Uniporter. And then MCU appears to be the pore-forming subunit. So now I want to dive a little bit into a bit of deeper mechanism on both of these components. So the first question is, what does MCU1 do? And this has really been the work of Kim Kamer, who again is an alumnus of University of Wisconsin. She's a 4th-year chemistry PhD student working in the laboratory. Right? So there's this long-standing observation that the Uniporter, it's a channel, but it's actually a calcium activated channel. So multiple old studies have shown that there's an external calcium binding site to the uniporter, and this external calcium binding site is important for the activation of the uniporter. And so MCU1 has these EF hands, and so it emerges as a reasonable candidate for mediating the calcium sensitivity of the uniporter. So we originally identified MCU1, and if you look in the genome, there's actually a total of 3 paralogous genes. It's one of 3 paralogous proteins. So there's a MCU1, MCU2, and MCU3. They all arose by genome duplication. MCU3 is only expressed in the CNS, so we've decided to focus on MCU1 and MCU2. Okay? So both of these proteins have a very similar protein domain organization. They both have mitochondrial leader sequences. They both have EF hands of the calcium sensing flavor. And so one of the first experiments that Molly Plavanich and Kim Kamer did was they pulled down MCU, again, the pore, and they could actually show that that pulls down both MCU1 and MCU2, suggesting that MCU1, MCU2, and MCU reside within the same complex. And moreover, if we actually pull down specifically MCU1 flag, we can actually pick up MCU2 as well. And so this clearly shows that they all co-IP each other. More recently, what Kim has been able to do, she's been able to express in bacteria and purify MCU1 and MCU2. She's worked hard on getting these protocols working properly. And when she mixes MCU1 and MCU2 together, and we try to size this using light scattering experiments, we see what appears to be a dimer. Okay? Now the challenge is MCU1 and MCU2 have very similar molecular weights, and so it's not clear if what we're looking at are dimers of MCU1 and MCU1, or if we're seeing dimers of MCU1 and MCU2, or if we're seeing a mixture of all of the above. And so to distinguish this, what we've done is we've done some differential scanning calorimetry experiments. We actually check to see what the melts look like. And so this is what the melt of just MCU1 looks like. This is what the melt of just MCU2 looks like. And then when we mix the two of them, we see a very different profile. And so this, in combination with our Malls experiments, strongly suggest a direct interaction between MCU1 and MCU2. We believe that MCU1 and MCU2 form direct interactions. They're forming a heterodimer. Okay. So what do these proteins do? And so there's a really important paper that came from Kevin Foskett's group, and then later from Yuri Hedjanotsky's group. We collaborated with Yuri, but this is really his paper. Both of these papers showed that a really important function of MCU1 is in gatekeeping of the uniporter. And so they're the ones that first showed that if you lose MCU1, or ordinarily if you give a small pulse of calcium to mitochondria, they actually exclude it. Okay? Remember, the old studies actually suggested that in order to get calcium in, you had to get a threshold spike of calcium on the outside. And what these two stories from the Foskett group and the Hedjanotsky group showed is that if you lose MCU1, they now will continue to drink this calcium even if it's not a big pulse. What Kim was able to show she was able to confirm this result using Talon knockout cells in which we knocked out MCU1 altogether. We also extended this gatekeeping function to MCU2 as well. So MCU1 and MCU2, if we lose either one of them, they will now allow small pulses of calcium to come in. Right? And so I'm going to show a little bit of data over the next 2 slides. I'm going to show you the model first, and then hopefully you'll be convinced that the model is correct based on the data. So our model is that MCU1 and MCU2 reside in the intermembrane space. They sense calcium, and if it's a small pulse, it's not going to get in. If there's a big pulse of calcium outside that exceeds a certain threshold, that high calcium concentration can now engage the EF hands of MCU1 and MCU2. This now will disinhibit the channel, and now the pulse can go through. Okay? This is our model, that these are operating in series, and that they're basically negative regulators of the pore. Okay? So let's see if we can garner additional evidence for this. If this is true, if this model is correct, then in principle, we ought to be able to create EF hand mutants of MCU1 or MCU2 that basically will lock the channel in the closed position. And this is exactly what Kim sees. And so she takes knockout cells. We knock out MCU2 or we knock out MCU1, again using Talon, so it's a complete knockout, and then reintroduce in point mutants. So these EF hands have exactly 2 point mutations in each of the EF hands. And when we express these EF hand mutants, as you can see, even when we give a big pulse of calcium now, it is not getting in at all. Okay? So this is what the knockouts, right, will transmit a big pulse of calcium, but these EF hand mutants are just locked in the off position. And so that helps to support this model that I just introduced. Another part of this model is that MCU2 requires MCU1 for interacting with the pore, whereas MCU1 does not need MCU2 to interact with the pore. So we have done a series of genetic and biochemical experiments to place these three components in a pathway, MCU, MCU1, and MCU2. And so I'll actually start with the biochemistry data first, and then we'll go to the genetics. And so the important point over here is that we can take MCU1 knockouts or MCU2 knockout cells, and then we express MCU1 flag or MCU2 flag. And if we do an IP, if we do a flag IP, what you can see is that this is the key band over here. We don't get MCU if we express a MCU2 flag and we try to pull down MCU. We don't pull it out if MCU1 is absent. Okay? In other words, if we pull down MCU2 in the presence of MCU1, we get MCU. However, if MCU1 is not present, MCU2 can no longer IP MCU. And this is not the case for MCU1. As you can see, there's bands in the other 3 or 4 lanes. And so what this means is that in order for MCU and MCU2 to co-IP each other, you have to have MCU1. And that's why we place it intermediate between MCU and MCU2. So that's the biochemical experiment that places MCU1 between these 2 proteins. We also did a genetics experiment. And in this genetics experiment, if we express the MCU1 EF hand mutant in the absence of MCU2, we can lock it in the off position, which means that the MCU1 can operate, the mutant can operate independent of MCU2. But as you can see, the converse is not the case. A MCU1, this MCU1 knockout, if you express a MCU2 EF hand mutant, it can no longer close the channel if MCU1 is no longer present. So that's the most complicated slide in the talk. Hopefully it made sense, and hopefully you believe this model. So our model is that MCU1 and MCU2 operate in series to gatekeep the channel. Okay, so these are threshold sensors, that confer AM signaling properties, if you will, to the Uniporter. Okay. So that was Kim's work, and that helps us to understand what MCU1 and MCU2 are doing. Now I want to get back to the pore-forming subunit. So at this point in the story, there's tremendous evidence in the literature that MCU is indeed the pore-forming subunit of the Uniporter. Okay? And remember, Albert Lehninger had shown many years ago that yeast mitochondria don't have Uniporter activity. And so if MCU is really the pore, we ought to be able to express human MCU in yeast and reconstitute Uniporter activity in yeast mitochondria. And so this is probably one of the most vexing experiments in our lab for the last couple of years. We could express human MCU in yeast mitochondria. It's expressed, we can detect it by Western blot. It's running as a higher molecular weight complex. However, when we give pulses of calcium, absolutely nothing is happening. Right? So this is the pore. Right? This is the pore-forming subunit. If everything that I've told you is correct, then we would expect there to be some calcium uptake. So this was really frustrating for us. And nobody else in the field could actually do a proper reconstitution using MCU as well. And so we're kind of puzzled by this. And if you look back at these BN pages, there's at least 3 known components to this band. But even then, even with our we thought that there might be something else that's lurking within this larger molecular weight complex. So Yasmin Sanchak, who's a postdoc in the lab, she was a biochemist trained in David Sabatini's laboratory. She's been a postdoc in the lab for the last 5 years or so. She came in and expressed a flag-tagged version of MCU stably in HEC293 cells. We grew up leader cultures of these cells and then did a flag IP. And what you can see is that when she IPs MCU flag the the IP migrates at the same molecular weight as does mitochondria from these cells. So this suggests that her IP is complete. We did a quantitative proteomics experiment, and we identified only 5 mitochondrial proteins that are very high scoring above background. So what do we see here? We see MCU, and it also has a paralog called MCUB. We picked up MCU1 and MCU2, which we've been talking about for the last few minutes, and we picked up a protein that had never been studied before, C22ORF32. It is a mitochondrial protein, and we ended up renaming this essential MCU regulator, or EMRE. Okay? And so what is EMRE? Okay? So EMRE is a metazoan-specific protein. Okay? It emerged, you're not going to find it in fungi. You're not going to find it in other exotic protozoa. You only find it in animals. It's a tiny protein, has a mitochondrial targeting sequence. It has a transmembrane domain, and it has a very D-rich carboxy terminus. By now, we've switched over from RNAi to using talins. And what you can see is that if we ordinarily give a pulse of calcium to permeabilized cells, they will uptake this. That's the normal uniporter activity. If we knock out EMRE using 2 different talins, there is no calcium uptake whatsoever. So EMRE is absolutely essential for uniporter activity. We did some topology studies that suggest that it's an N in, C out protein. Okay? And so EMRE is pulling down with the uniporter complex, which we call uniplex. And if we knock it out, we lose uniporter activity altogether. Okay? So this is one more complicated slide, but I want to walk you through it. So we were able to make knockouts of not just MCU, but also MCU1 and EMRE. Remember, this is what the uniporter complex migrates on native gels. If we lose MCU, we lose it altogether. What you can see is that in the absence of MCU1, this complex migrates at a different molecular weight, and that molecular weight resembles what happens when you lose EMRE. Okay? So this led to the hypothesis that what EMRE was doing was bridging MCU, the pore, with this regulatory machinery. And that's shown here nicely. This is an IP experiment that's performed either in wild-type cells or in cells in which we've knocked out EMRE. Okay? So what we're going to do is we're going to express MCU as a flag fusion, and we're going to pull on it. And if we pull down MCU, if we pull that down, we get all the components of the uniporter complex. We get MCU1, MCU2, MCUB, and EMRE. Now, if we only eliminate this one protein, okay, we tug on this, what's going to happen is we still pick up MCUB. Okay, so these two are still together. However, MCU1 and MCU2 are now gone. So what this suggests is that EMRE is playing at least 2 functions. First of all, it's absolutely essential for calcium uptake through the pore. And then secondly, it's very important for the association between MCU1 and MCU2 with the membrane-associated components of the uniporter. Okay. So this is what we think the uniporter complex looks like right now. And so I've told you that MCU and now EMRE are both necessary for calcium transport. And now we decided to go back and see what happens if we express both of these proteins in yeast. And when we do this experiment, we were super excited when we saw this. And so now, if we only express MCU in yeast, we purify their mitochondria, they do not uptake calcium. If we express MCU and EMRE, what you can see is that they now uptake calcium and buffer it. This is now sensitive to Route 360. You need to have phosphate, you need to have a membrane potential. So this fulfills all of the classical properties of the uniporter. So we've now been able to achieve a reconstitution of the uniporter in yeast mitochondria, which are basically like test tubes for the uniporter. So I told you that EMRE is a metazoan-specific protein, but that seems to be at odds with the fact that you find the uniporter across multiple branches in eukaryotic tree of life. Okay? And so just one other thing that we've been able to do recently is to trace the evolution of MCU, MCU1s, and EMREs. And so humans, right, have all of these proteins. They have MCU and MCUB, they have 3 MCUs, and they have EMRE. Yeast are like test tubes for the uniporter complex. They don't have any of these proteins. Now, these are both what are called the opisthecons, and then the out group includes amoebozoa, things like Dictyostelium. So the Dictyostelium genome has an MCU and a MICU, but no EMRE. Okay? And so I just told you that we need MCU and we need EMRE to reconstitute uniporter activity in yeast, but here's an organism that doesn't have EMRE. It's an animal-specific protein. And if you look at Dicty MCU and Homo sapiens MCU, the domain organization is very similar. They both have mitotargeting sequences. They're different here. They have a coil-coil domain, transmembrane domain, a dime motif, transmembrane domain, and a coil-coil. And so they look kind of similar based on sequence. However, when we express DDMCU on its own, that is actually sufficient for calcium uptake as well. So what we think happened was that MCU and MICU1 were part of the earliest eukaryotes. If you look on the eukaryotic tree of life, these two proteins were probably present in the earliest mitochondria. And then during evolution, yeast, as well as other fungi, decided to dump this apparatus altogether, and Homo sapiens actually evolved a need for EMRE. We think that EMRE helps to keep MCU in an open conformation, and it allows it to be regulated by MICU1 and MICU2. And just as additional evidence that DDMCU is kind of a special protein, we can take human cells that are knocked out for MCU or for EMRE. They do not uptake calcium at all, but the Dicty MCU protein can complement either MCU or it can complement EMRE. And so this is kind of a special protein. It kind of works on its own. And so this is our current model of the uniporter complex. We call this the uniplex. I've told you that MCU is the pore-forming subunit. In humans, this probably assembles in a closed conformation, and the expression of EMRE probably keeps it open. EMRE also allows this pore to now be controlled by MICU1 and MICU2. So this is our current model for how this works. And so we're really excited because now we're sort of entering into the molecular era of the uniporter. This is something that had been studied extensively in the context of metabolic coupling and signaling and cell death, and now we know the molecular machinery. And over the last couple of years, the field is moving extremely quickly. Now that we know the molecular components, a number of groups have been able to either RNAi or overexpress these components. And at this point, this is a table from a review article that we're writing right now. The uniporter complex has now been linked to oncogene-induced senescence, to cell migration, to neuronal excitotoxicity, to the inflammasome. It's actually becoming difficult to identify a cellular process that is not linked to the uniporter, which has its own dissatisfaction, but it's linked to a lot of different fundamental cellular processes. And the other exciting thing that's happening now, and this is not work from our lab, by the way, this is other work, and others, not us, have also reported what happens to organisms or even humans where uniporter components are mutated. And so Torin Finkel's lab at the NIH had a beautiful paper where they characterized the trap mouse, the knockout mouse for MCU, and on a mixed genetic background, it's actually viable, and it has exercise intolerance. It's a very small mouse, and so it's been exciting to see that particular paper. And mice that have lost MICU1, they die 1 or 2 days after they're born. We're actually working up those mice now. We're working with Cricket Seidman's laboratory on the MICU2 mice. These have a very interesting cardiovascular phenotype. And a neuromuscular lab in the UK reported just last year the first human patients with mutations in the uniporter. These are patients that have mutations in MICU1 that have a skeletal muscle myopathy. Their OXPHOS activity is totally normal, and they also have extrapyramidal signs and some learning difficulties. And so in the next few years, it'll be really exciting to see just how this channel complex is relevant for human disease. And we're also very excited about leveraging the power of evolution and evolutionary diversity to really try to understand the molecular mechanisms by which this channel operates. And so I'm going to close now. And so I've shared with you how our laboratory is using the mitochondria inventory to study both human disease as well as basic biology. And just one final sort of approach that we're trying to develop to try to decipher the function of all these proteins. We're trying to develop new computational tools that will complement some of the amazing biochemical studies that are happening here and genetic studies elsewhere. And so as all of you know, there's a lot of genome sequence that is now being deposited into the public domain. We have lots and lots of even eukaryotic genomes that are becoming available in the era of next-gen sequencing. There's also a ton of microarray data that's in the public domain. And I like showing this slide. So Young in our laboratory produced this slide. So this basically shows the number of Affymetrix and cDNA microarrays that are in the public domain. And as of 2013, there are 1.3 million microarrays that are in the public domain, which corresponds to about 26 billion measurements. And this is billions of dollars of data that's in the public domain that all of us can just download and analyze. And so several members of our laboratory are now trying to develop computational tools, developing methods very similar to the search engines that underlie Yelp and Netflix. These are programs that, based on your past movie selections, will recommend to you new movies that you might like based on the statistics of many other individuals that reported that they like movies similar to the ones that you like. So we're developing some new tools that leverage the power of all of this freely available data to look for signatures of co-evolution or co-expression to try to shed insight into the function of your gene of interest. And so if you're interested in these tools, we've served this method up at a website, geneclimb.org, and we're developing a similar tool, a Bayesian tool called ClimbX, that will integrate over many microarray data sets to provide functional clues to your genes of interest. And so with that, I'm going to stop. I've already acknowledged most of the current members of the laboratory that are working on the Uniporter complex. I want to give another shout out to the former members of the laboratory that made really important contributions, both to build a mitochondrial proteome, as well as some of the early studies of the Uniporter complex. These are some of our collaborators, and these are our funding agencies. So with that, I thank you again for the invitation to come and speak. And I'm happy to take questions.
Where does MCU fit in these? Where does an MCU fit in the broader ion channel family? Does it have a P loop? Does it have any molecules of ACSAs and the other?
Yeah, no, great question. So it actually doesn't bear any resemblance to any known ion channels. And this is probably one of the reasons that it was never identified. Based on primary sequence, it doesn't look like anything. It doesn't look like ORI, for example. I would say the closest thing that is perhaps the core A, the magnesium channel. Core A is the bacterial magnesium channel. It's known as MRS2 in mitochondria. And that also has two transmembrane domains flanked by coiled coils. But obviously, there's no high resolution structures right now, so we don't know how it's doing its business yet.
You showed that there's a point mutation in MCU1 that's related to RE360 sensitivity, but that RE360 also inhibits EMRE. So do you have an idea of what this point mutation is actually doing?
Yeah, so the S259A mutant is in the MCU. That's actually the pore-forming subunit. So that's going to be right at the entrance to the pore. So we actually think that RE360 literally plugs up the pore. It's positively charged, and we think that the serine residue might somehow be important for the interaction directly between it and RE360. And that's why when you mutate it, we no longer get the inhibition. Well, we have not been working on the lipid composition of different. I think it's a fascinating question, but it's above my pay grade. Yeah.
What's the link to the energized mitochondria?
I'm sorry. What's a?
Since it only passes calcium when the mitochondria is energized.
Yeah. That's a great question, actually. So you only get calcium uptake when there's a voltage. So then the question is, what happens in depolarized mitochondria? And so Albert Lehninger, during the final 5-6 years of his career, he was actually studying the Uniporter. He actually hypothesized that all this calcium that gets accumulated, later on it can actually get secreted as well. And so there's actually data that suggests that when the mitochondria are depolarized, you can actually get calcium efflux through the pore as well. So these might also be sources of calcium as well.
Okay, I think to kind of keep things on time, we might hold off any more questions for now. But let's thank Vamsi again for a great talk.